Good morning to one all present here. Hope your day has been pleasant so far. I am Sharanya Dumre Patil,
And I, Abhishek Tiwari,
will be your host for today. So let me begin by saying that it is a privilege and pleasure to welcome you all on behalf of Amity University Mumbai to this webinar on KIT signaling pathways in cancer progression and treatment by Dr. Maheshwaran Mahi Sir, organized by Amity Institute of Biotechnology AIB and Center for Drug Discovery and Development CD3. This is an especially important event as tomorrow, 4th of February, is observed as World Cancer Day. The day is aimed at raising awareness about the global impact of cancer and the need for greater efforts to prevent, detect, and treat the disease. It is an opportunity for individuals, organizations, and governments to show their support for the fight against cancer and take actions to reduce the number of preventable cancer deaths. Hence, webinars like these play an important role not only in bridging the gap between the scientists and students, but also inspiring future researchers.
Now this reminds me of a poem from the early 1900s called Miss Chi by W.H. Auden. An extract from it goes like something like this. Cancer's a funny thing. Nobody knows what the cause is, though some pretend they do. It's like some hidden assassin waiting to strike at you. Childless women get it, and men when they retire. It's as if there had to be some outlet for their foiled creative fire. We have come a long way from this, but we have so much more left to discover.
That said, I would like to introduce Dr. Penna Suprasanna Sir. He was the former head of the Nuclear Agriculture and Biotech Division at BARC Mumbai and former professor at Homi Bhabha National Institute, Mumbai, India. He has recently joined Amity University, Mumbai as Dean Research, Professor and Director of Amity Center for Nuclear Biology and the Head of Institute of Amity Institute of Biotechnology. Now, I would request respected Suprasanna Sir to give the welcome address. Over to you, sir.
Yeah, a very good morning to everyone. Honorable Chancellor Sir, Honorable Vice Chancellor Sir, Professor A.W. Santosh Kumar Sir, and Dr. Vinod Prasanna, Dr. Vinod Kannan, and the guest of the day, Dr. Maheshwaran Mani, and you both who have nicely organizing the event for today. Very good morning to everyone and welcome to you all. As already said, World Cancer Day is a global initiative which started in 2000 and then it's going on ever since. And since last year, we had Close the Care Gap. That was the campaign which started last year. So this being the second year, I think we are into more creating an awareness about the world cancer problem that is going on and the initiatives that are taken to improve quality care, screening, early detection, treatment, and palliative care. So there is a lot of progress that is needed in understanding more about the mechanisms, how we can treat better the human population. And there are 6 key areas that are coming in cancer research. Prevention, early detection, treatment, palliative care, surveillance, and so on. So I'm very happy that CD3 of AIB has initiated this World Cancer Day program. I feel very delighted to be a part of it. As they say, create a future without cancer. The time to act is now. I think we have an eminent speaker with us who will talk about much more about the cancer research. Thank you and welcome to you once again.
Thank you, Suprasanna Sir. Now I would like Abhishek to continue.
Abhishek, you are on mute.
I would like to welcome Professor Honorable Vice Chancellor, Professor Dr. A.W. Santosh Kumar Sir. Honorable VC Sir has 33 years of academic and administrative experience and has held the post of Vice Chancellor for a period of 4 years in one of the highly ranked universities. Previously, he worked in the United States for a period of 16 years and has held various positions such as Dean, Academic Affairs, Accreditations, Co-Director at Inland STEM Consortium, Co-Director at Center for Genomics, Director at STEM Program, and Professor of Microbiology and Infectious Diseases. The Honorable Vice Chancellor, sir, completed his PhD from the All India Institute of Medical Sciences, New Delhi, and further completed his postdoctoral fellowships from the University of California, National Cancer Center of Singapore, J. Craig Venters Institute, the United States of America, National Institutes of Health, the United States of America. His primary research focus includes the study of human microbiome role in systemic diseases, which is funded by the NIH USA. His other area of research is the host pathogen interaction and drug discovery that is funded by DBT and DST India. He has published 181 peer-reviewed research articles, 4 books, 12 book chapters, and has 18 patents. His academic services include sitting members in various extramural funding agencies nationally and internationally, and also is currently serving as editor and editorial board member of many high-impact peer-reviewed journals. I request Honorable Vice Chancellor, sir, to give the opening remarks. Over to you, sir.
So good morning, everyone. Respected Dean of Research, Dr. Suprasanna, the esteemed invited speaker, Dr. Mageshwaran Mani, my dearest faculty members who are pioneers from different alma mater, my dear student friends, the most elite 2 compeers here. It's indeed a very great pleasure to inaugurate this webinar on commemorating the World Cancer Day. And many people we know, we are not a novice that we know about cancer, and more than 68% of the world research focuses only on cancer in biology. And this is like chasing a chicken every day in the morning, where we just move on to chase a cure, and the cancer gets permutated into different means and arms, where diagnosis is a very big problem, treatment is a very big problem, and ultimately there is a tussle between life and death, where always death succeeds. But in between, God has given a lot of brain, energy, and work that each and every scientist, biological scientists, are quite fascinated, especially our students are quite fascinated about cancer and study of cancer. And cancer, as you know, that when you talk about Indian statistics, 100 people in a population of 1 lakh is affected with cancer by one way or other. And this data is getting extrapolated year by year. And we just focus and become the 5th on the leaderboard in the top 5 countries which has the most incidence of cancer, where China and the USA, they top the leaderboard. So having said that, commemorating the World Cancer Day is always remembering one day of a very great episode which totally mars one's life into miserable ideations and sequences. So we biological scientists are involved one way or other, and I'm indeed very happy that Center for Drug Discovery and Development, we popularly call as CD3. And as you know, there is a coincidence for CD3 because CD3 is a T-cell receptor activator, which focuses on the helper and the suppressor T-cell, which combats cancer as well. So this is very adept that CD3 is now bringing about this initiation of the function of commemorating the World Cancer Day in 2023, which is none other than bringing action into a paradigm shift. And every year there is a theme that is given. And so making action publicized is one of the important themes in this year to combat cancer. So we look forward to a lot of researches. We look forward to a lot of interests that are coming in cancer. There are a lot of cures that are coming in cancer. And India has now moved on to a new paradigm shift, which you call it as Ayush, which is an ethnomedicine based thing, where plant-based alkaloids and other things are now currently being used for immunotherapy and cancer. So having said that, I don't want to stand in between this invited speakers talk and you all. I look forward to having a very good deliberation of this World Cancer Day. And I again congratulate the CD3 and the AIB team for putting forth this function. Thank you very much.
Thank you, Honorable Vice Chancellor, sir. We are glad that you took time out of your extremely busy schedule to be part of this event. Now, I request Sharanya to introduce our speaker of the day, Dr. Maheshwaran Mani. Over to you, Sharanya.
And this brings us to the main part of this event. I would like to introduce the speaker before he begins the talk. Dr. Maheshwaran Mani, sir, had done his PhD in microbiology from Justus Liebig University in Gießen, Germany in 2006, followed by postdoctoral fellowship in immunology and stem cell biology from Stanford University, California, USA in 2011. He specializes in immunology, stem cell biology, and medical biology, and has received numerous awards, including 2022 Poster Award Jury from India EMBO course, CCMB, and the Best Teacher Award from Pondicherry University in 2019, as well as in 2022. He was also the recipient of 2019 EMBO Research Leadership Course Travel Award from the Wellcome Trust CBT India Alliance, among other things. And he has completed several research projects and has patent in modulation in KIT signaling. He also has several publications in internationally reputed peer-reviewed journals. I would request sir to deliver his talk, and I would also request the participants to send their doubts and queries in the chat box. It would be better if we have an interactive session. Over to you, sir.
I'm audible?
Yes.
Yes, sir.
Okay. Good morning to all. Dear Vice Chancellor, dear Dean, faculties, students, I thank you all for giving such a wonderful opportunity to present our research work, what is going on in our laboratory for past 10 years. Just give me a moment, I'm going to share my screen. Okay, so today for the day, actually I thought of discussing with all of you about a piece of work, what is going on in my laboratory for past few years. That is a molecule that is c-KIT, we also known as KIT. So this is a molecule and it has a multiple nomenclature. Some laboratories, they call this as a c-KIT. Some of them they call as a KIT. And it also has other nomenclature like stem cell factor receptor, still factor receptor, and mast cell growth factor receptor. So it's a molecule which is called by multiple nomenclature names in different laboratories, but all means the same. So just to start with today, since I come to know that there are a lot of audience at the student level, especially undergraduates and postgraduates. So instead of getting into data, I thought it better to give some background or some fundamental information and then jump into the ongoing research, what is happening in our laboratory as well as worldwide. So today I'll be focusing on a couple of things. First, I will be talking about cell surface receptors, and then I'll be discussing with all of you about receptor tyrosine kinase family, and then I'll be talking about c-KIT, which is a part of this receptor tyrosine family. I'll be giving some information about what is structure, what are the signaling pathways that are regulated by KIT, and what are the functions. And then I'll be moving on with different diseases that are associated with KIT, and what are the KIT-associated cancer progression. And finally, I'll be landing with how we make use of KIT signaling as therapeutic targets to treat different cancers. So this is the open talk. So before getting into the line, so we all have learned from the textbook about cell surface receptors. So there are different receptors, that is ion channel receptors, G protein coupled receptors, and enzyme linked receptors, right? So these are, we will be learning it from the textbook for biochemistry. And if you look into the enzyme linked receptors, so again, there are different enzyme kind receptors. One is receptor tyrosine kinases, then receptor serine threonine kinases, and the tyrosine phosphatase kinases. So there are different enzyme linked receptors. And the c-KIT, which the topic of the day, it belongs to the receptor tyrosine kinase family 3. So again, if you look at the background, so this receptor tyrosine kinase family, they have a specific property. Some of the receptor tyrosine kinase family, they have all the tyrosine kinase family has. If you look into the intracellular domain, so all what you see here is the intracellular domain. They all have a tyrosine kinase domains. So that's the uniqueness of all the receptor tyrosine kinase family. However, there are some unique difference. That's why which make them as a receptor tyrosine kinase family 1, like RTK1, RTK2, or RTK3. So if you look some of the receptors, tyrosine kinase, they are rich in cytosine-rich domains. Some of the RTKs, that is receptor tyrosine kinase, they have a extracellular domain, and they have a intracellular domain and transmembrane domain. And what there is, you see there is a disulfide bridges which connects the transmembrane domain with the extracellular domain. So this is the one type of family of receptors. And there is a receptor tyrosine kinase family 3, which is where the c-KIT also belongs into that. What is uniqueness of this receptor is they have a extracellular domain, and this extracellular domain, they have an immunoglobulin-like domain. If you want to see here, this is there are 5 domains: domain 1, 2, 3, 4, and 5. So this has 5 immunoglobulin-like domain, and this is unique of receptor tyrosine kinase family 3. So one example is platelet-derived growth factor receptor, and even c-KIT also belongs to this receptor tyrosine kinase family 3. So just to give a full view, so what we see is the different receptor tyrosine kinases there. If you look at here, and this is the receptor tyrosine kinase family 3, where the c-KIT also belongs to that. So along with c-KIT, there's a colony factor, stimulating factor receptors, platelet-derived receptor factors. So there are a lot of receptors which belongs to this family, and c-KIT is one type of receptor that belongs to receptor tyrosine kinase family 3. And with this background, actually, I'll be focusing into the c-KIT pathways. So just to give you an idea, the c-KIT is a receptor tyrosine kinase. It belongs to receptor kinase family 3. And what is unique is it has immunoglobulin domain, and it has kinase domain. So this is a very important property of c-KIT. So as I told you, it's a membrane. c-KIT is a transmembrane glycoprotein. It belongs to class 3 receptor tyrosine kinases. So that's why I told, like in the beginning itself, this receptor, it's also known as with different names. That is KIT, stem cell factor receptor, mast cell growth factor receptor, and still factor receptor. If you get into literature, every laboratory, they'll be having their their style of addressing this molecule. In our laboratory, we address with the KIT. So in some laboratory, they address with the stem cell factor receptor, and some laboratory they address with the still factor receptor. So, but however, doesn't matter, all means the same, and they all are the same molecules. Okay, fine. And this is the receptor, and the ligand for this receptor is stem cell factor. Okay, before getting to the picture, actually, I want to give again one more time. This is a c-KIT. It has 5 immunoglobulin domains. It has a transmembrane domain, and it has a kinase domain, right? So this is the structure of c-KIT. The ligand for this receptor is stem cell factor. So that is a cytokine. And even the stem cell factor, also there's a different names. Again, some laboratory, they address with the stem cell factor. Some of them call as the KIT ligand. Some of them call this a still factor. Okay, so the ligand for the receptor is stem cell factor. And this ligand, they are mostly expressed in fibroblast, and they are also expressed in endothelial cells. And this ligand, they are present in 2 forms. One is in membrane bound form. If you look at here, so this is a cell membrane. So what you see, the cartoon, this is a cartoon that represents the stem cell factor, that is a ligand for c-KIT. So this ligand, it is expressed in 2 forms. One is in a soluble form, that you see here, it's a soluble form, and other one is a membrane form. So what happens is, the ligand is expressed as a membrane form, and there is a cleavage site. What we see here, that's a cleavage site, and this cleavage site, there is a proteolysis that happens, and upon proteolysis, the extracellular domain they got cleaved, and they act as a soluble cytokine or soluble ligand. But the cells also express a membrane bound, what we see here, which lacks the cleavage site. So that means they are all the time present in the membrane. So there are two types of stem cell factor. One is a soluble factor, and another one is a membrane bound stem cell factor. So this is a ligand which binds to the receptor, and the binding of ligand is very much essential for the receptor to activate and do signaling transduction. So what we see, there was a model proposed. We all know, again, this is c-KIT. So if you take a cell, the cell has a KIT receptor, and usually they are present in a monomeric form. So what you see here, this is a monomeric form of c-KIT. So c-KIT is in a monomeric form. So if you take any cell which express c-KIT, they are in monomeric form. So they have monomer, they have monomer, they have monomer, they have monomer. And whenever there is a ligand gets inside, this receptors, they start sensing the ligand. Okay, so what happens here? So their receptors are in monomeric form. They are in monomer, they are monomer, and they are monomer. So when there's a ligand, the monomer starts sensing, and this induces the dimerization of the receptor. So what you see here in this picture, in the second one, you see the two monomers, once they sense the ligand, they are coming towards and they become a dimer, right? So what you see, the third stage, it's that dimerization of the receptor. So once the ligand comes, they start sensing the ligand, they get dimerized, and once the receptor get dimerized, what happens? There is a conformational changes takes place in the receptor, and upon the conformational changes, there's a kinase domain here, there's a kinase domain here, and these kinase domain, they go for autophosphorylation of the receptor. So if you look at the receptor, there are many tyrosine residues are there, and these tyrosine residues are phosphorylated by its own kinase domain. So it's an event. The receptors are in a monomeric form. Whenever the ligand comes, they get dimerized, and once they get dimerized, what happens? The kinase domain phosphorylates. We call it the autophosphorylation. The kinase domain, it phosphorylates the tyrosine, and once they got dimerized, what happens is, if you look at here, again, this is the kinase domain, and this is the kinase domain, and you see there are multiple tyrosine residues. There is a residue 568, 570, 703. It goes all the way till 936. These are different amino acid positions. So we have 1, 2, 3, 4, 5, 6, 7, and 8. So there are 8 tyrosine residues in this molecule. So once they get dimerized, what's going on says, like all the tyrosines are phosphorylated, and once it's phosphorylated, now it is inviting the downstream effector molecules, which is at the cytoplasmic level. So it is inviting all the molecules. So what you see here is the effector molecules, which is interacting with the c-KIT, and upon interaction, what happened? It transmutes the signal. So what is signal? The stem cell factor is a signal which goes there. The receptor receives the signal. Receptor get dimerized, get activated. It phosphorylates. Once it phosphorylates, all the molecules at the downstream, they are binding to the receptor. What you see here is binding here, and after binding, they get activated. Now the signal is transduced. Now the signal transfer. What happens next? It's going to happen like this, right? So this is the c-KIT. So ligand binds. Once the ligand binds, what happens is they get phosphorylated. Once they get phosphorylated, all the molecules are the effectors. They're interacting with c-KIT, and they're transducing, and they end up with activation of multiple signaling pathways. Look at here, c-KIT is activated, and upon activation, they activate SOS, and they're finally activating the RAF pathway, and that activates ERK, and finally it is end up with cell proliferation. Look at here, the c-KIT also activates JAK-STAT. Okay, the JAK is associated with the KIT, so the JAK activates a STAT, which is a signal transducer. Okay, and it's a transcription factor. And once the STAT is activated, they go all the way to nucleus. They induce gene expression, and this signal is reported earlier to be important for cell differentiation. And it's also there as pathway where it's a Lin-Fin pathway, which activates the RAC and J and K, and this also end up with proliferation. Similarly, if you look at, there are also other effector molecules, AKT or mTOR pathways, and these pathways are also activates NF-kappa B pathway and downstream pathways, and they also involved in cell growth or cell survival. So look at this molecule. It's a molecule which receives signals, it activates, and it transduces signals to downstream effector molecules, and which keep activated. And finally, what we're having is either cell proliferation, or differentiation, or survival. So this is what happens with the c-KIT. And this mechanism is not new. It is studied for last 2 decades. The KIT is very old. People are studying for past 20 to 25 years. So it is a well-studied molecule. But what comes to interesting thing is the unnormal conditions, they are doing the cell proliferation, differentiation. But sometimes what happens, there is a mutations. So the KIT undergo mutations. And when the KIT undergo mutations, there is an imbalance. So what they have studied is, there is a mutation which takes place in intracellular domains of the c-KIT, especially in the kinase domain. When there is a mutation, all the signalings are not going in a regulated manner, but they are going unregulated manner. And because of this unregulated manner, what happens? It is excess activations or lack of activations. And excess activation of KIT signaling end up with the cancers. And when there is a lack of signal, it end up with developmental defects, right? So what I'm going to show here, okay, before that, this is just information out of the box. So the KIT signaling molecule, as I told you, it is a very important molecule, which is necessary for cell proliferation, survival, differentiation, right? So there are lots of study has been done, as I told, for past 25 years. And this signaling has been shown to be very much important for hematopoiesis, where all the hematopoietic cells are produced. They are also reported to important for melanogenesis. They are important in melanin formation, and they are also involved in gametogenesis. So people who are working KIT signaling, they are not only focusing towards cancer. There are laboratories focusing KIT signaling in melanogenesis. People who are working with KIT and focusing gametogenesis, and people who are working with KIT, they're focusing gametogenesis. And what we do in our laboratories, we are focusing mostly on hematopoiesis and see how these molecules are regulating hematopoiesis, right? Fine. So now, if you look at here, as I told you, it's a very important receptor. And now this receptor, it has 2 things. Whenever there is a lack of this c-KIT function, it is end up with a huge problem. Whenever there's a gain of function, it has another problem. Look at here. Whenever there is a mutation loss of KIT, or even there is a mutation loss of KIT ligand, that is stem cell factor. Whenever there is a loss of KIT or KIT ligand, it ends up with abnormalities in hematopoietic system, nervous system, or reproductive system. Indeed, there is lack of development. So it is very important. And people have studied in the early stage. There are many investigators. What they did is they were trying to produce a knockout animal of c-KIT. Okay, they're trying to knock out a c-KIT and produce a knockout c-KIT animal. And what they observed, this is the embryonic lethal. So it is very hard to produce a KIT knockout mouse. So that is a huge problem. So people are trying for long, but they couldn't able to do it. And later, there is an investigator from Cornell University in New York. What they have done it is actually, there's a group from Tellini. They have developed a mouse model. Instead of producing a knockout animal, they have produced a reporter system. Okay, so it's a little different task of producing KIT animal. So that's one thing happening in the subject. So KIT signaling, if there is no KIT signaling, when there is a lack of KIT ligand, it has a huge developmental defect. So there is a lack of hematopoiesis, lack of nervous system, and lack of the reproductive system. Okay, fine. Now what happened if the mutation? If there is a mutation, and because of mutation, if there's excess activation, it ends up with cancer. And so far, several cancers has been reported, which has because of excess activation of KIT, we call it a gain of function. And some of the reported cancer is GIST, that is gastrointestinal stromal tumors. There are also cancers reported in hematopoietic systems, in germ cells, and in mastocytosis, which I'm going to show in the next slide. Okay, if you look at here, so far what I have discussed with you is I discussed with you what is receptor tyrosine kinase. I was just talking with you about what is c-KIT, how the structure looks like. I was discussing about the ligand, how they look like. Then I was discussing you about the pathways. What are the different pathways like a RAS-RAF pathways, PI3K pathways, JAK-STAT pathways. So what are the different pathways are involved? And I also explain you like when there is a lack of KIT signaling, we have developmental defect. And when we have a excess activation or gain of function, we have a cancers, right? So as I told you, in our laboratory in Pondicherry University, we are trying to study this molecule in terms of hematopoiesis. So what you see here is a hematopoiesis. So where in hematopoiesis, it's where all the blood cells are produced. So what you see at the bottom is the T cells, B cells, NK cells, all granulocytes, macrophages, mast cells, erythrocytes. These are the blood cells, or else we call as mature cells, right? So if you look at here, there are stem cells. This is a long-term hematopoietic stem cell. And what you see is multipotent progenitors, common lymphoid progenitors, and common myelin progenitors. So why I have shown this is, I have discussed with you what is c-KIT. I told what happens if there is no KIT signaling. I was to explain you what happens if there is an excess KIT signaling, right? So now next thing comes to the picture is, which are the cells that express this KIT receptor, right? So KIT receptor is expressed in all the stem cells. Just keep in mind that like all the stem cells express c-KIT. Okay, fine. Then how about hematopoietic stem cells? Yes, hematopoietic stem cells also express the KIT. And look at short-term hematopoietic stem cells, they also express c-KIT. Multipotent progenitors, they also express c-KIT. And if you look at CLP, they express but less in number. And if you look at CMP, they express less in number. Now, if you look at all the progenitors, they all express. And coming back to the adult cells like T cells and B cells, there is no expression. Look at T cells, there is no expression. Look at B cells, there is no expression. However, interestingly, there are two cells. One is mast cells, another one is megakaryocyte. Again, they are expressed. So that means this is a receptor which is not expressed in all the cells, but in stem cells as well as few mature cells like mast cells, megakaryocytes, and somewhat in NK cells, right? So that's what's happening is worldwide, people are trying to study the c-KIT signaling molecules in each and every cells. People are trying to understand the signaling molecules in stem cells. People are trying to understand signaling molecules in mast cells, megakaryocytes, and so on, right? So this is the picture to explain you that what are the cells that express the receptors. Now let's get into the picture of KIT signaling in cancers, right? Just 1 second, guys. So as I said earlier, the KIT signaling is very much essential. When there is no KIT signaling and there's a lack of KIT signaling, that's a developmental defect. When there is an excess, there is a cancer. So what does it mean? So we need a regulated or well-balanced KIT signaling mechanism in a healthy individuals. So having excess activation is a cancer. With no signaling, it is ended with a developmental defect. So several studies have been done in the past, and what they observed, whenever KIT signaling is activated in excess, it ends up with cancers like GIST, that is a gastrointestinal tumors. It ends up with the melanoma. KIT signaling, having excess KIT signaling also end up with a small cell lung cancer, carcinoma. There are also reports that KIT signaling also excess activation end up with testicular carcinoma and mastocytosis. This is like having excess mast cells. And when there's excess mast cells, they end up with the mastocytosis, then asthma and allergy, and goes on like that, right? And KIT signaling excess activation also reported to cause acute myeloid leukemia. So we have, it's not one single cancer, guys, okay? So it's not one single cancer. We have like leukemia, we have a solid tumors and lot of carcinomas. So which makes this receptor is a very important receptor to investigate and go for this receptor as a therapeutic target to treat these cancers. So I will give you a couple of other examples how these receptors are involved in different cancers. So okay, if you look at this image, the one what you see at the top is a c-KIT. This is what I started in the beginning of the lecture. It's a structure of a c-KIT. But you see there are 5 immunoglobulin domains, 1, 2, 3, 4, and 5. This is 5 immunoglobulin domains, which I discussed in the beginning of my talk. And this is a TM, that is a transmembrane domain, which is in the transmembrane region. And what you see here, this is a kinase domain. So we have a kinase domain 1 and kinase domain 2, right, which is in the cytoplasmic region. And what actually they have done is they have did a clinical analysis. This is a clinical analysis done with the GIST, whoever had a gastrointestinal tumor, and the patients who are referred to clinic. So what you see, this is a graph. What you have in y-axis is the number of cases, and what is in x-axis is the mutation which is reported in the which part of the c-kit, right? So what this is a clinical data. So what they have done, whoever has reported with the GIST, they collected the samples and they did a sequencing, and what they observed is in all the patients who are reported with gastrointestinal tumor, they found the mutation is in a kinase domain. You look at here, for most of the cases, what is reported, you see here, the mutation is in kinase domain, in kinase 1 region. And there are a lot of second cases. You see there is some of the cases in kinase 2 regions. So this tells us whoever reported with the gastrointestinal tumor, when they were doing a sequence analysis with those patient samples, they found all the mutations were happening in the kinase domains, which few examples in the extracellular domain, but majority of the mutations were happening in the kinase domain. So what does it mean? So whenever there is a kinase domain mutation, so now once the kinase got mutated, usually what happens to c-kit is it is a homodimer, so it's a monomer. When the ligand comes, they become dimer, the kinase get activated, it induces signaling. But in this mutation, what happens when this is the mutation, they don't need any ligand. So without ligand, they are going for constitutive activation. So once the mutation happens, the KIT signal is continuously activated. We call it a constitutive activation, and the constitutive signaling mechanism end up with the continuous cell growth, and the continuous cell growth end up with the tumor. So this is what happens here. Look at this slide. So this is our next slide. So there are cases where there is a mutation in a KIT region, kinase domain region, and because of that, the KIT acquired a gain of function. It has an excess function, and that gain of function, it end up with thymic carcinoma. It end up with a gastric tumor. And look at here, mastocytosis. There is a mutation here where at 5-6 region, there was a mutation, and that also end up with when I talk about 5-6, it is a 5-6-0 is the position of amino acid in c-KIT receptor, right? So if you look at the 5-6-0 mutated, that end up with the gain of function, and that causes mastocytosis. And there was also reports of melanoma and thymic carcinoma at 5-7-6 region. And there is also acute lymphoblastic leukemia. If you look at here, there was a mutation at 642E region, and this mutation ended up with the gain of function, and that causes acute myeloid, acute lymphoblastic leukemia. And there are also acute myelogenous leukemia where the mutation is at 816 position, and that end up with missense, and which has a gain of functions, and it causes multiple cancers. So here, what this tabular column summarizes is most of the kinase domain, if it get mutated, it end up with gain of functions, and that gain of functions end up with multiple cancers like thymic carcinoma, melanoma, GIST, mastocytosis, or acute myeloid leukemia. And still there are a lot of this is a recent review paper. Okay, still what you see here, there is a mutation, but there is still this mutation are yet to be characterized because the effect of protein is not known here, right? So which tells us that the c-KIT mutation end up with a gain of function, and that gain of function end up with the cancer. So still long way to go. Still, even though this molecule is studied for last 2 to 3 decades, still a lot of functions are not fully studied, and still there are a lot of laboratories which is investigating the molecule. Okay, so I like this image always, so that's why I thought of showing to these young students. So this is, if you look at the year, it is 1915. That's a mouse which was captured and reported in 1915, and these are the black mouse. Okay, and this is a black mouse, and you see the white patches, and later we come to figure out that why they have this white patches is these all the mouse which has received the mutation KIT signaling molecule, KIT receptor, and because of the mutation KIT receptor, they end up with a lack of melanogenesis, and there was lack of migration of melanin to skin region, and that's why this mouse has these patches, even though it has not much to deal with today's topic. This is the mouse which has mutation in KIT molecule, and because of mutation, there is a lack of melanogenesis, and because of that, you see there's a patches, right? So these are the examples of KIT. So there are lots of KIT. As I told you, KIT knockout is not possible because they are embryonically lethal, but a lot of KIT mutant mouse are there. So a lot of KIT mutant mouse are there, and if you look at this KIT mutant mouse, The wild type mouse are black in color, but if you see the mutant, they are brown in color. Sometimes they have some patches. You see a lot of differences. Just by morphology, you can differentiate and tell, oh, this could belongs to the KIT mutant mouse. Okay, look at this picture again. As I told you, it's not only KIT signaling molecule is reported to be involved in carcinoma or GIST or myeloid leukemia. Even there are a lot of cases reported where the KIT expression is associated with breast cancer. So what you see, the figure A and B, these are the tissue of breast cancer, and the tissues were labeled with the KIT molecule and they captured image. What you see in the pink is the KIT expression, right? So the A and B, what you see in the pink color, these are the KIT expression in breast cancer cells. And what you see on the C and D is the negative control, where you don't see much of KIT expression. So this is a histology which shows that how the KIT expression is more. And there's a paper which is suggesting that the KIT expression is also associated with breast cancers, right? Fine. Okay, this is the next paper that again they have done with breast cancer samples as well as with the lung cancer samples. They have done with they have analyzed the serum sample as well as the tissue samples, and what they have found is here this little they have went with in-depth analysis, and they've also told how this galectin is involved in KIT associated cancer. And this is just another piece of paper to show that the KIT signaling molecules important. What you see here is the KIT, and the KIT is also involved in breast cancers as well as in tissue cancers. Okay, fine. So with this, actually, I will just put a pause for KIT associated cancer, because it is a ocean. If I keep going, there's not a one single cancer which is caused because of KIT. The story continues. So I just put a pause there, and I want to also give a small add up of what is happening with KIT, because this is a very important receptor molecule. If you look at here, this is a KIT receptor where the ligand binds, induce signals. And what our lab as well as some of the labs have reported is this KIT is not only activates and induce signal. We have also reported that that this KIT also activates the other receptors. We call this as a cross receptor. So if you look at here, the KIT ligand binds to the receptor where the receptor is activated, and this activated receptor, it activates other receptors, and it is inducing a different signaling molecules. And this mechanism is also important to do a cell-specific functions. So if you look at here, there was early studies that the KIT activates the erythropoietin receptors, and they are involved in erythroid differentiation. And our lab, we also reported that KIT activates interleukin-7 receptors, and that is important for thymic differentiation. And recently in 2021, for our lab at Pondicherry University, we also reported that the KIT activates interleukin-4 receptors, and which is important for hematopoietic cell proliferations. And why we focus on this, because this is also crucial, because this cross receptor interaction mechanism also contribute for differentiation as well as the multiple or excess proliferation. Okay, so this is extra additional data which we thought of doing it. So now let's get into the next, the last section of the today's topic, that is therapeutic targets. Okay, if you look at here, this is receptor tyrosine kinase family 3. This is what I started with, RTK3, where it's not only KIT which belongs to RTK3, however, also like there are other receptors like the platelet derived growth factor receptors. They also belongs to receptor family 3. Fine. So this is the RTK family 3, and so far I have discussed what happens when c-KIT is activated and what are the different cancers that are associated with c-KIT. I also discussed with you what happens when there is no c-KIT. There is a development defect, right? So whenever there is excess activations, we get cancer. Okay, whenever there is a lack of KIT, that end up with development defect. So this receptor tyrosine kinase is extremely important, and so whenever there is a imbalance in this signaling mechanism, it ends up with a lot of malignancies, and this makes this receptor as an excellent target to go for therapy target. So what happens is there are a couple of ideas what people are practicing worldwide. As I told you, actually, the c-KIT receptors are existing as monomer. Once the ligand binds, they become a dimer, and that dimer, it goes for auto phosphorylation, and it induces a lot of downstream signaling molecule. Whenever there is a mutation, even without ligand, it get auto phosphorylated and it ends with cancer. So now we want to make use of this molecule as a drug target site and develop a drugs for cancer. So a couple of targets are there. What they do is actually they develop an antibody which binds to this molecule, and once they bind, they block the heterodimerization, right? So that is one method of developing a drugs against the c-KIT. So developing a antibody that blocks the dimerization. So that's the one method. So there are also other antibodies that are produced which go and bind to the ligand, and it neutralizes the ligand. Once the ligand is neutralized, these neutralized ligands cannot bind to the receptor and it cannot activate the receptor. So that through neutralizing antibody of ligand, that drugs can be targeted. That is one method. And there are also other anti-receptor tyrosine antibody developed, where the antibody go and bind to the receptor, and which will block the antibody binding, and through that signaling can be blocked. That is other method. And there are also lots of drugs are developed where they are targeting the kinase domain of the c-KIT. So what happens? The kinase is activated and it phosphorylates. So they are trying to develop a lot of kinase inhibitor. So these kinase inhibitor go and bind to the kinase domain and make sure that they are not activated, and it blocks the downstream signal target. So this is a concept, and this is what happens. And based on this concept, lots and lots of drugs has been developed so far in the market. Some of them are already in practice, and some of them are in clinical trials, and some of them are still in the research phase. So if you look at here, this tabular column summarizes a list of drugs that are targeted, not only for c-KIT, but for all the RTKs. Look at here, imatinib. That was the, in fact, inhibitor which was developed to target KIT. And in fact, this is approved and it is in practice. Actually, people who are having a KIT-associated cancer, those patients are taking imatinib because this is already approved. It's already in practice. And a lot of companies are marketing, and one company is Novartis. And look at the other inhibitors like SU11248. So this is the another compound which is indolin 2-1. So this is in clinical development. There are a couple of other compounds like PKC targeting and midostaurin. This is also in development, clinical development. So there are lots of other drugs like MLN-CT535. Okay, that's a culoleucine compound. So this is also KIT based drugs. It's also clinical development. So these are a few examples because I just pulled out this data from a review paper. However, it's a 2014 paper, but you could see a lot of developments are going on. How people are trying to develop drugs to target KIT to recover from the KIT-associated cancers. And this is a timeline. What we see here is starting with 2001 to 2021. You see the approval of first kinase, and that is imatinib, was happening in 2001. So it is in 2001 the first approval was given for imatinib, and now officially imatinib is given for treating common myeloid leukemia, and imatinib is also given for treating gastrointestinal tumor cancer. Okay, so imatinib is approved drugs people were giving for treating KIT-associated cancers, and followed by lots and lots of inhibitors has been developed over the period of time. If you look at here, there is a inhibitor developed for VEGF receptor. There are a lot of inhibitors developed for downstream targets. So this is a huge milestone. People started focusing on these pathways and trying to develop drugs. And some are in development, some are in practice, and some are in research phase. So what you see here is a little new report where they were trying to work out with developing multiple compounds, and they're trying to develop the targets to treat KIT mutations, which is happening here. And in fact, they are also trying to develop for GIST, right? So this is a lot of happenings developments are happening for KIT-associated cancers. And this is again a report where there's a drug called dasatinib and radotinib. This is also developed to target c-KIT, and it's not only imatinib, which is already in practice. there are some drugs like dasatinib and radotinib, which is also in development to treat acute myeloid leukemia. Okay, fine. So to start, so this is what I have done so far. So to quickly summarize, this is a c-KIT, and this is a very important receptor. The excess activation causes cancer. Lack of KIT causes development defects. So now this becomes extremely important biological receptor molecule to focus, so that a lot of drugs have been developed to treat KIT-associated disease. In all the early days, actually, the drug targets were focused on either the extracellular level or the intracellular level. But there are some challenges. All these drugs, they are not only highly specific for c-KIT, for example, imatinib or all this dasatinib. It is not only focusing on blocking c-KIT, it also has some side effects. It is also trying to block other molecules and gives a unspecific mechanism. So what is the research happening or going on worldwide at current moment is, people are not only trying to focus developing targets for c-KIT, they also try to target based on intracellular molecules at the downstream targets. So people are trying to develop JAK inhibitors. People are trying to develop STAT inhibitors or ERK inhibitors. Like that's what I told you in the slide. If you look at go back here, you see there is a somewhere I have seen here actually. See, they have MEK inhibitors, right? So in 2020, they are developing a inhibitor for MEK. Okay, so a lot of the drugs are keep it like in practice, so that to go for a specific treatment for KIT-associated cancers, right? So I'll stop here, guys. I'll summarize what I have discussed. I think I'm running out of time, so I will just quickly summarize. So today I started with discussing about what is cell surface receptors, and then I was discussing with all of you about receptor tyrosine kinases, and I discussed about how the KIT looks like, what are the KIT signaling mechanisms, and what are the KIT-associated diseases. Then I talked about KIT-associated cancers, and I talked about how KIT can be potentially used as a therapeutic targets. So the take-home message is two things. KIT is a very much important receptor, and excess activation causes cancer, and lack of activation causes development defects. And the second take-home message is KIT could be a potential therapeutic targets, and it is not only KIT, even the KIT dependent, the downstream effector molecules like RAS-RAF pathways, JAK-STAT, all these downstream could be also be used as a potential therapeutic targets to develop drugs, so that this can be given for KIT-associated cancers. Right. So I will stop here. I thank you all. This is just a picture of Pondicherry University, which is snapped by one of our students, and it is circulating. Whoever is going and presenting, they circulate this image. I thank you all. Thank you so much for patiently listening to my talk, and I really thank the organizers for providing such a opportunity to share my data with you all. Thank you all. Thank you so much. And if you have any questions, I'm so happy to take the questions. Thank you all.
Thank you so much, sir, for this wonderful talk. It was really very insightful and attention-grabbing. Yes, the audience has a couple of questions, and with your permission, I would like to ask them on their behalf. A student has asked the question, how to treat a person if they are c-KIT positive? Is there a possibility?
How to treat a patient if they are c-KIT
positive?
Okay, fine. Okay. Keep in mind that when you use the word c-KIT positive, as I told you, it's a receptor. What we use as positive is when the cell express, we call it positive. When the cell don't express, we call it negative. So that's why we use the term. Almost everybody is KIT positive, because if we take a healthy individuals, all their stem cells have c-KIT. Okay. So the question is actually, I would put the other way around. It's not that they are KIT positive, they're getting cancer. The KIT is doing a lot of functions. Instead of doing a less function, they're doing a lot of functions or gain of functions. So they end up with the cancer. So that's why one of the approved drug is imatinib. So what they do, we have to give imatinib to the patients, and through the drug, the patients can be slowly recovering from the KIT-associated cancer. So there are drugs available. One drug is imatinib. So give the imatinib over the period of time, and just see how the patient recovers. Okay. So that is the answer, right? Yeah.
So one last question that we have is, you mentioned galactans. So can you explain them in very brief?
Okay, fine. This is a mechanism. What they have studied is actually it is not only happening at the extracellular level that where the ligand binds to the receptor and get activated. So there are so there are a lot of intracellular molecules. So which also regulates this excess KIT activations. So they have found that the galectin plays a role, and wherever they have seen the galectin, they have seen that like the KIT associated cancers also fluctuating. So that's how they connected the data of. It's not our data. I just pulled from literature. So they have studied from the serum sample, and they found that there is a relevance of galectin in KIT cancer, right? So it is a mechanism what happens at the downstream targets, which regulates the KIT associated.
Thank you so much, sir.
Can I have? I have a question. It was a very fascinating, very interesting talk to learn about, KIT on the involvement in different types of cancers, and you also spoke about the drug. So, is there any drug resistance that is coming up in this due to some mutation?
Yeah, that is there, sir. Yeah, actually, if you go back, actually, one second, just a moment. I hope my screen is visible to all of you.
Yes.
Look at here. This is imatinib, right? And it's already clinically approved. It is approved, and then it is in practice. But this imatinib is a kinase inhibitor, and it is not only binding to c-KIT, so it also binds to other kinase inhibitor. Sometimes what happens is actually a couple of things. We are targeting for a particular molecule, but it may also go and bind. See, because these are sequence-specific inhibitors, they go and bind. There are similar sequence with other molecules. So when they go and bind unspecifically, that also end up with a problem. So you might be targeting a cancer patient, but they are having problems with other developments. So this is what we see as a drug resistance. Okay, so that's why. So instead of seeing as a drug resistance, we see as an unspecific reaction of the drug. So that's why people are still focusing to go for specific targets. As of now, when there is no drugs, then we have to go for it. But people are trying to focus with the downstream targets instead of focusing only the KIT. Go for downstream targets, and that downstream targets are giving little specific blockage of KIT's excess signaling rather than at the higher level. So these are the problems we are challenging. Challenges happens in treating these patients.
So as I understand, the drug resistance and then non-specificity are two different things that you do. So the drug resistance is due to some mutations that occur in KIT?
Yeah, again, fine. Good. Actually, I forgot to mention
something
that.
Like secondary mutations.
Yes, exactly. Because if you go back, if you remember the initial slide which I showed, it is not in one mutation. Because of mutation, this the KIT acid cancer is happening, right? So there are multiple mutations. Like what you told, a patient is having a mutation in one particular domain, say for example, kinase 1 domain, and he is given target. By the time he is having treatment, there are possibilities he might acquire mutation in other domains also. And in that case, the Clevac or imatinib may not work for him. So yes, I agree with you. The secondary mutations also end up with drug resistance. So in that case, what we have to we have to go for sequencing, and then we have to go
more complexity.
And then we have to go for alternative therapy, and that's why we are not ending with one compound, and people are still keep screening for multiple compounds. What happens? Sometimes what they do, sometimes they go with instead of single inhibitor, they put multiple inhibitors. Okay, so instead of one, go for two. Then that gives a high chance of recovery. So these are the challenges. That's why we aren't having endpoint for this kind of treatments. We go for treatment, but we can't see completely coming back. There is still always some effects will be there.
Thank you, Dr.
Yeah, thank you, sir. Thank you.
With this, we've come to the end of the event. And now I would like to request Dr. Vinod Prasanna, sir, an assistant professor at Amity Institute of Biotechnology, to deliver the vote of thanks. Over to you, sir.
Thank you, Sharanya. Can you hear me?
Yes.
Thank you so much. And on behalf of Center for Drug Discovery and Development and Amity Institute of Biotechnology, Amity University, Mumbai, we would like to extend a very hearty vote of thanks to Dr. A.W. Santosh Kumar, Vice Chancellor, for his constant encouragement and support to conduct this event. We would like to extend our thanks to Speaker Dr. Makeshwar Mani for his wonderful informative talk. I would like to render my sincere thanks to Dr. Suprasanna sir, Director AIB, and Dr. Vinod Kannan Ravichandran, CD3 in charge, and Dr. Nilesh and Dr. Shashank, the members of CD3, for organizing this event smoothly, and the students volunteers, Mr. Abhishek and Ms. Saranya, and the faculties and the students for attending this seminar. We are also grateful to our social media officer, Mr. Chetan, and the IT team for their work and support. Thank you so much, everyone, for your time. And the session was very quite insightful and motivating. Thank you, sir. Thank you, everyone.